Hello, and thank you to each and every one of you for joining us on this momentous mRNA Day. I'm Brian Neel, COO here at TriLink BioTechnologies, and it's a great privilege and great pleasure to welcome you to a tradition of celebrating mRNA research inspired by the grand opening of our manufacturing facility and corporate headquarters in San Diego just 2 years ago. We're all here today virtually and across time zones, from our offices, labs, kitchens, and backyards, because of a shared passion for the rise of mRNA. Already felt across the globe, this passion has created a united energy to realize the full potential of mRNA technology across therapeutic areas. For that, and on behalf of TriLink BioTechnologies, thank you for your strong resilience in research. We are at the forefront of a scientific revolution, and all of us are fundamental to this paradigm shift. Before we get started, I would like to also express my sincere appreciation to all of you who have helped bring this event together. Thank you to our superb lineup of speakers, our skilled hosting partners, spirited attendees. Today will be fun and full of learning and celebration.
Thank you so much, Brian, and hello everybody. A very warm welcome to mRNA Day, an exciting virtual event brought to you exclusively by TriLink in association with GEN, Genetic Engineering and Biotechnology News, and the CRISPR Journal. We're broadcasting live from the East Coast, from New York and Washington, D.C., and you are among more than 2,500 people from around the world who've registered for today's exciting event and celebration. So hello again, I'm Kevin Davies, Executive Editor of the CRISPR Journal and the author of Editing Humanity, The CRISPR Revolution and the New Era of Genome Editing, which has got RNA in it. So I do hope you'll take a look at that book. mRNA Day is brought to you exclusively by TriLink BioTechnologies, as you've just met, which has made free registration today possible. So we really are very grateful to them. TriLink is making a very generous donation today on mRNA Day to UNICEF USA, and specifically to the efforts of ACT-A, that stands for Access to COVID-19 Tools Accelerator. ACT-A is a global collaboration that seeks to accelerate equitable access to COVID-19 tests, treatments, and vaccines. And on behalf of every registrant leading up to today, TriLink is making a donation in excess of $25,000. So thank you so much for your participation, which has made that donation possible. In fact, here is a short video about that effort from UNICEF.
No one said it would be easy. Delivering 2 billion vaccines to the world, COVID is still spreading and new strains are making the news. So we're not stopping. Together, we've reached over 130 countries so far. But we've got billions of doses to go because no one is safe until we're all safe.
We will get our program started in just a few minutes. I just want to set the stage, tell you a little bit about the hosting partners, and give you a few other helpful pointers as we embark on the next 4 hours. Jen, of course, one of the co-hosts for today, is Genetic Engineering and Biotechnology News, celebrating its 40th anniversary this year, covering the biotech industry almost from its inception, offering a free monthly magazine, which you can sign up for, daily news on biotech and the life sciences, an array of digital events and virtual summits and conferences. We also invite you to check out our new premium subscription channel, Gen Edge, which offers in-depth exclusive coverage of the business of biotech. You can find those stories and more information at the Gen website. From the same parent publisher, Marianne Liebert Inc., the CRISPR Journal debuted in 2018, publishing original research, reviews, and commentaries on all aspects of CRISPR biology and genome editing technology and applications. Edited by Rodolphe Barangu at North Carolina State University. The CRISPR Journal, you can find more information and submit papers at CRISPRjournal.com. And speaking of new journals, we are absolutely ecstatic to announce the launch of Gen Biotechnology in early 2022, a marquee new peer-reviewed journal. We've just announced the brilliant Hannah El Samad, a professor at University of California, San Francisco, as the journal's chief editor. And there's still time for you to be part of the debut issue, which will be coming out in just the next 2 or 3 months, We are accepting submissions, so you can find about the editorial board and the whole scope and purpose of the journal at genbiotechjournal.com, www.genbiotechjournal.com. So on this mRNA Day, we are excited to celebrate a magnificent period of mRNA research over the last couple of years, one that has literally impacted people all around the world and saved many lives. Over the next 4 hours, you'll hear from some 10 great speakers on all aspects of mRNA biology, vaccine development, and therapeutics. So here's how today will work, briefly. We'll begin with a plenary program here on the main stage that will run for just a shade under 2 hours. In a few minutes, we'll open with a keynote presentation from Linda Marbán. Then we'll have a virtual discussion featuring Norb Pardy and Mohamed Gabriel Alameh discussing mRNA vaccine history and applications, moderated by my colleague Juliana Lemieux. And we'll close the plenary session, taking us to the halfway point of today's proceedings, with a talk from MIT's Daniel Anderson. At that point, we'll split into 2 parallel tracks, track 1 and track 2. You can see the helpful navigation tabs on the left-hand menu, offering you more choice to hear speakers in your particular field of interest. Track 1 is entitled mRNA for Infectious Disease, and will feature talks from Andy Giel, Kiat Ruxrungtham, and Evelina Angov. And track 2 is What's Next for RNA, featuring Leo Zangi, Greg Newby from the Broad, and Jaspreet Khurana. Both tracks will conclude with a 15 to 20-minute panel discussion featuring most of the speakers in those specific tracks. And we will take your questions, which you can enter in the Q&A box you'll see on the right-hand side of your screen at any time. After a short break following those 2 tracks, we will return to the main stage for our closing plenary session that will commence at about 3:35 p.m. Eastern, 12:35 p.m. Pacific time, featuring a special presentation from Yasuhiro Furuichi, one of the legendary names in mRNA biology. And we will end proceedings with a short video, closing up at 4 o'clock Eastern, 1 o'clock Pacific. A few housekeeping items before we get the program underway. If you experience any technical issues, we know these glitches do come up from time to time, please refresh your browser. That's usually the best way to avoid any issues. Or you can send one of our event managers a direct message through the chat on your screen. The navigation menu on the left side of the screen is fairly self-explanatory, allowing you to move from the main session to either of our 2 breakout tracks at the appropriate time. You'll see there's a networking feature on that menu bar. You can request a chat with a specific attendee, or you can be paired up with another mystery attendee, sort of a potluck, I suppose. And also, please, if you have time, check out the Resource Center, where you'll find a wealth of useful information about TriLink, about CleanCap, how to contact the company, as well as COVID information from UNICEF and information from the other hosts of today's meeting, Jen and CRISPR Journal. If any of your colleagues or friends are unable to tune in live today, and we realize that for many of you in Europe and Asia, it's getting pretty late in the day, don't worry, mRNA Day is being recorded and will be available in its entirety on demand, starting in about 1 week's time, and will remain accessible for a full 12 months. So time issues are not a concern. And if you would like a certificate of attendance for today's mRNA Day, please email your request to webinars@libertpub.com, webinars@LIEBERTPUB.com, libertpub.com, and we will be happy to send you a certificate of attendance, which we know is important to many of you. Throughout the event today, you'll see a number of interesting poll questions popping up, along with some mRNA fun facts and, I'm told, TriLink trivia. We'd love to get your feedback on these poll questions. That could be very helpful. You'll see a visual cue asking you to click the poll tab to answer these questions as they come up. And in fact, I think the first one is coming up right about now. Okay, let's get the program underway. Our keynote presentation is from Linda Marbán, who is co-founder and CEO of Capricor Therapeutics. Linda earned her PhD from Case Western Reserve in cardiac physiology and continued her training at the Cleveland Clinic Foundation and Johns Hopkins. After a spell at Exogen, overseeing the development of gene therapy products, she joined Capricor, which is developing precision cell and exosome-based therapeutics for a range of diseases, including Duchenne muscular dystrophy and COVID-19. Hello, Linda. Great to see you. I'm going to pass it over to you.
Thank you so much. And thank you for asking me to be the keynote speaker for this very important day. As I'm booting up my slides, I'll sort of make a reference to what has become one of my favorite television shows of all time, which is Ted Lasso. And for those of you not familiar, Ted was invited or hired to coach a British football team in the Premier Leagues, coming from the American football tradition. And he felt like he was a bit of an imposter in a space that he didn't understand well, but yet did it to great success. And I'm hoping to have that same luck as I talk to you today about our experience with building RNA-based therapeutics and using exosomes as their delivery vehicle. My name is Linda Marbán, as Kevin just said. I come to biotech from the bench, and I work for Capricor. We're a publicly traded NASDAQ-listed company under the symbol CAPR, for those of you that are interested in reading our filings with the SEC. So let's talk a little bit about the history of the discovery of RNA and its implications and meaning for what we're doing currently with RNA. So RNA was discovered sort of as a byproduct of the discovery of the genetic code DNA. And for most of the last, let's call it 30 or 40 years, we've sort of thought of RNA as really the maidservant to DNA. It took the code given to us by the DNA, transcribed it faithfully, translated it, transported it, and ultimately led to its expression of all of the proteins that are responsible for life. This early discovery was made in the early 1960s. So it's been a journey of nearly 60 years to the understanding of what RNA actually does biologically. And all I can say is that I believe that the future of medicine perhaps is held in this very important nucleic acid, RNA. We now know a lot more about RNA. We know that it is not really just sort of the maidservant, as I said before, to DNA, but in fact actually plays an important role in biologic regulation. Transcription and translation has many flavors and many directions, and RNA is the mediator of all of those complex processes, so to speak. So now maybe DNA is taking a step back from the forefront as sort of just holding the code, so to speak. In any case, RNA is divided into two basic subtypes, that which codes, mRNA, which will be the focus of what we talk about today primarily, and then non-coding RNAs, and those are being discovered as rapidly as the papers can be published. And they're broken down into several categories that we know now, primarily antisense oligonucleotides, microRNAs, siRNAs, and aptamers. And I'll touch on each of these categories with the exception of aptamers a little bit as we go through the talk today. But as I mentioned, we'll be mostly focusing on that which is coding. So as I mentioned, it took a long time for us to begin to understand the power of RNA. And we look at the first seminal experiments and then published by Wolf and his colleagues in 1990. So remember I said RNA was actually discovered in 1961. So you're talking multi-decades before the first published work using or demonstrating the fact that RNA can be used independently of DNA. And what they did is they took mRNA, they injected it into skeletal muscle, and they were able to demonstrate the expression of the protein. The control group was that they took DNA, they inserted DNA by the same direct injection method, and were also able to see expression of the protein. So this basically demonstrated that you could use RNA independently of DNA and derive a high fidelity functional protein. Now, where does this take us in terms of the therapeutic utility of RNA? Well, we're already seeing it today with the vaccines, but it really has opened the door, this general concept that we can use RNA for a variety of biologic uses to actually attack or prevent diseases. We can alter protein coding, We can change or inhibit protein translation. That would be what those siRNAs I spoke about a little bit ago, and could be extremely important in managing oncology downstream. And we can also use RNA with the specificity to target binding so that we can actually have expression of proteins where we want them, as opposed to Wolf and his colleagues who were left with the idea that you had to inject it directly into the site where you wanted the message to be delivered. Why is the use of RNA so intriguing? Why have we worked so hard to understand it? Because you can lead protein expression without integration into the host genome. So that means you don't have any of the risks of downstream expression or off-target expression or oncogenesis or any of the issues that come to mind when we think about, for instance, CRISPR technologies, which we certainly are intrigued about as a general concept of modification of gene expression. And of course, currently available gene therapies, which use viral vectors, which have significant safety implications from using that very important vector, but one that can actually lead to the immune system being highly activated. So let's talk about the development of RNA therapeutics. What happened between 1990 and 2020? That was nearly 30 years from the first attempt to use mRNA as a therapeutic. Why did it take so long? Well, there's a variety of reasons, and nearly all of them are ones that, with the community of science behind us, we have come to the fore to be able to address, and now are sitting on the cusp of the real excitement, which is using RNA to treat diseases, and in fact is actually occurring as we speak around us. So what happened in that 30 years of development? Well, first we had to deal with the inherent instability of RNA. Obviously, our body is trained to recognize RNA as dangerous. Many viruses are RNA viruses. So when RNA comes into your body unprotected, your immune system is activated and gets rid of it very quickly. You can't deliver very easily to target tissues. As we saw with the early studies, you had to directly inject it into the site that you wanted expression. We're now working on all kinds of targeting technologies and delivery strategies, probably the biggest hurdle the field is facing and has had to face to get it to where we want it to go. The toxicity sort of ties back to the immune aspects of it. You know, you put a lot of free RNA in the body, it does not like it, causes cytokine storm and a variety of other unwanted immune responses. And of course, effectiveness has always been an issue, because if you can't get it where you want it to go, you can't get it to express, you can't really get it to be effective in treating different types of pathologies. Now, all of that has been under active development and experimentation over the past 30 years. And so within the last 5 years, we've had 10 RNA products approved by the FDA. And I think I actually have read that there are several more that are coming to be approved in a very short period of time. So RNA therapeutics are now really becoming part of the standard pharmacy that will be available to treat a variety of diseases. How did that occur? Well, the most important one was modifications of the RNA. Some of this has actually been pioneered by our host today, TriLink, who have worked very hard to make RNAs that are more stable, resist degradation, and are able to have high fidelity protein expression. And then, of course, delivery. Robert Langer, one of the well-known, we'll call him fathers of biotechnology, and many of the discoveries that we have in our warehouse of ideas today, has always stated that we have the hardest time getting past the cell membrane. And once we can do that, once we can improve drug delivery, the whole field will open up, as I believe it is doing right now. So how is RNA delivered? Well, currently there are multiple ways that it can be put inside the body. And please forgive this busy graphic. I promise I will walk you through it, and you'll understand it at least at a 30,000 foot level by the end of the slide presentation. So at the top of the human being, shown on the left here, you can see that two ways of delivering RNA are in the eye: intravitreal and subretinal. Now, we tend to think as a field that the eye is perhaps one of the best places to test the new concept of RNA or nucleic acid delivery. It's a protected space, it's a capsule, so you can deliver small amounts of your therapeutic and derive a big biologic impact. It is largely not vascularized, and it is immune privileged. So it is a wonderful testing ground, sort of a primordial pond, so to speak, in which we can put the therapeutic in, and ultimately see if the biologic effect we are trying to derive is in fact going to occur. So very popular place for RNA delivery, and probably the most advanced in terms of therapeutic products. The other ways, intrathecal, intravenous, subcutaneous, and intramuscular, all are still sort of harking back to that concept, with the exception of intravenous, of trying to get the contents of your therapeutic to where you think it should be going. The most important of these, obviously, in terms of commercially approved products, is intrathecal, where we've had 2 approved products, neither of them directly mRNA. Antisense oligonucleotides is the major product, where we are treating the disease spinal muscular atrophy, we being the field, not Capricor or us, obviously, and to great effect. This is a disease, spinal muscular atrophy, where children are born with the inability to move. Most of the children die from respiratory failure prior to the age of 2, and have never even been able to roll over or sit up. With the use of these game-changing, revolutionary biologics using intrathecal delivery of the RNA, these children are sitting up, walking, and living. Absolutely one of the most astounding scientific discoveries and product development experiences in modern history, I do believe. Subcutaneous, intramuscular are again trying to get the RNA to the site of where we're trying to treat the disease. It works, but it's obviously cumbersome. Dosing is difficult. Bioactivity is hard to measure, and long-term product stability is one that will have to be explored further. Intravenous is the gold standard. We're all working towards that. We want to be able to treat systemic diseases by intravenous delivery of RNAs, and ultimately that will come over time. As of right now, there are no approved products using intravenous delivery. Shown here on the right are the types of delivery paradigms that are being developed. These antisense oligonucleotides, by far the most impactful in terms of product approval. These block areas on the gene so that protein transcription can occur without taking into account a mutated section. You get a fairly functional full-length protein with the mutated section sort of X'd out. And these therapeutics seem to be biologically active, if not long-term effective, which we'll see over time. siRNA, I mentioned briefly, this is a way of blocking protein transcription so that you can typically shut down the biologic activity of the cell. It's being explored specifically for oncology indications, and there's a lot of promise there. Lipid nanoparticles are being used to deliver all of these potential concepts, whether it be an mRNA or an siRNA. A lipid nanoparticle is what's being used by the current vaccines, and I'll talk more about them in a few minutes. And then if you want to get directly to the gene, currently available gene therapies, you have to use a viral vector. The most popular right now is AAV, adeno-associated virus. This allows integration into the genome for long-term protein expression. But as I mentioned, there are safety implications that have prevented this technology from taking off with the speed and with the success that one would hope we would be able to have. Shown here is the list of approved RNA therapies that I've alluded to. What you can see just by scanning this slide is that most of them are these antisense oligonucleotides. And to the eye were the first attempts. Some of these have been discontinued. The most active in the space in terms of the antisense oligonucleotides are for spinal muscular atrophy, as I mentioned, and also Duchenne muscular dystrophy. The only 2 approved mRNA products as of right now are the vaccines for COVID, one by Moderna, one by Pfizer. I think we're all aware of those and how they have catapulted the field forward in a most extraordinary way with this intramuscular injection of a lipid nanoparticle coated mRNA for the spike protein to drive an immune response to COVID-19. And I'll talk a little bit more about that in the next few minutes. So let's switch gears for just a brief moment and start talking about the real issue in RNA delivery, which is delivery. How do we get those RNAs into the body to overcome those boundaries and barriers that I spoke about a little bit earlier? So the currently available technology is to use lipid nanoparticles. They used to be called liposomes. They have had their contents modified and changed over time. We've come to understand that they have to have a certain amount of polarity, that they have to be cationic lipids involved, that they protect the RNA, and that they allow it to be delivered. And this is sort of, stand up and cheer because we can get it inside the body, but not so fast, because lipid nanoparticles can be toxic. They cannot easily be targeted. We haven't seen any evidence yet in any clinical indication of targeting of a lipid nanoparticle. And they may drive hypersensitivity. Most of them contain an ingredient called polyethylene glycol, kind of like antifreeze. It actually is used in many biologic products, but in some people it does drive a hypersensitivity response and may also be potentially responsible for some of the implications that we've seen from the vaccines, such as myocarditis. And I'll talk about that also in the next few minutes. We at Capricor are pursuing a different strategy. We are taking advantage of nature's delivery system, which is called exosomes. I'll be talking a lot about exosomes in the next little bit. So this is the preview or the teaser. Let me just tell you that exosomes are very natural. They are nature's delivery system. As I mentioned, a simple blood transfusion contains billions, if not trillions of these. And as you know, don't therefore drive an immune response. We've seen, and others have seen, that they are non-immunogenic. You can multiply inject them over time into at least animals and don't see an immune response. They can be targeted. You can put moieties on the outside of them so you can tell them exactly where you want them to go. And they naturally deliver contents inside the cell, allow us to cross the cell membrane and attack that problem that I mentioned very early on, which is getting biology beyond the cell membrane. So now I'm really going to dig in and talk to you about exosomes as delivery vehicles for genes and other therapeutics. So what exactly is an exosome? I alluded to their bioactivity in the previous slide, but I didn't tell you what they were. Exosomes are a class of extracellular vesicles. These are made by the endosomal processing pathway. They are nanometer-sized lipid bilayer vesicles that are rich in RNAs and proteins, and they are secreted by nearly all cell types. As I mentioned, you can find them in blood, you can find them in milk, you can find them in beer. Pretty much any biologic fluid is packed with exosomes. They are good for driving biology. They are how cells communicate with each other. I'm going to talk a little bit in the next few slides about how they were discovered and what they did. But basically what I can tell you is the aha moment was recognizing that cells need a way to communicate with each other. They need a way of transmitting materials between them. And this is what exosomes do. They are actually nature's delivery system and therefore offer broad potential for therapeutic applicability. So exosomes were discovered in the 1980s. They were considered to be the trash can of the cell and sort of considered to be like a lysosome or some other type of small extracellular vesicle and sort of cast to the curb. Some smart scientists about 20 years later opened up the exosomes and found that the contents inside did not speak to trash. It spoke to communication. As I mentioned, RNAs, proteins, lipids. And so it began an exploration into exosomes. And ultimately, as you can see from the number of publications shown in the upper left-hand graphic, the field is exploding. The graphic ends at 2019, but I can assure you that 2020 and even 2021 will have even more publications suggesting that exosomes are becoming the forefront of the development of potentially not only biotechnology, but also science itself. Exosomes are the smallest of the extracellular vesicles. They typically are sized at 40 to 100 nanometers. As I mentioned, they are lipid bilayer vesicles, and they contain biomolecules of protein, RNA, DNA, and lipids. And what you can see from this graphic on the left, and I'll highlight this in the next slide a little bit more, is they actually have receptors on their surface that allow us to take advantage of that in delivering contents inside the cell, but they're natural receptors that allow ligand binding so that they can provide signaling to the cells themselves, deliver the content inside of the cells, and drive changes in protein transcription, translation, and a variety of other bioactivities. So how did we discover our exosomes? As Kevin mentioned in his brief introduction, we actually had been working in cell therapy for a long time. We have a late stage product to treat Duchenne muscular dystrophy. It's an allogeneic cell product. We worked on that product for a good 10 years, and we were really scratching our heads as to the mechanism of action. We knew that if we, for instance, took a mouse or a pig or even a human that had had a heart attack and we injected in the cells, we would see biologic impact. We would see reduction in scar and improvement in cardiac function 6 months or even 12 months later. And we really started scratching our heads because the cells themselves disappeared. They were gone. They were gone, in fact, after a month. So we now have a situation where the cells are not persisting. The therapeutic benefit is maintained long after the delivered cells are gone. They see endogenous regeneration kickstarted. We see all kinds of opportunities for that with activation of endogenous cell cycle activity and driving a creation of new cells, whether it be in the heart or skeletal muscles, specifically with our cells. And we knew that paracrine signaling played a role in that, but as most of you know, paracrine signaling is typically gone after about a week. So we actually started a deep dive into what made the cells work. And what we found was that they were releasing exosomes that was driving the bioactivity. So shown here on the bottom are two figures taken from our seminal report, which was published by Ibrahim and his colleagues and Stem Cell Reports in 2014. And what we were able to show is two very important things, and I'm going to call your attention to the graphic on the right first. So we took a group of mice, we gave them all a heart attack, and then we did several scientific manipulations of them. The first group were injected with exosomes made by our cells. Our product is called CAP-1002, but the scientific name for it are CDCs, cardiosphere-derived cells. So we took cardiosphere-derived cells, exosomes. We took a normal human dermal fibroblast exosome, comes from a simple fibroblast, and we took PBS, and we injected the mice on the same day with all three of these constructs. So each group got one of the constructs. And what you can see is that the CDC exosome group had an improvement in ejection fraction that's similar to what we've been seeing over 10 years in using the cells in the mice. We did not see the same benefit at all in the fibroblast exosome group. They had the same response as the PBS injected mice, which is that their ejection fraction continued to worsen. Okay, so now we think, right, these exosomes might be driving biology, but we have to actually see if we take them away, will we still have the effect? And in fact, what we were able to show in this study, where we took the cells now, we took the CDCs themselves, we took one group of cells and we just treated it with a vehicle, DMSO. We took another group of cells and we treated them with GW4869, which is a ceramide inhibitor. We took the mice, same day, all mice had a heart attack. One group got the control, the DMSO. The other group got the GW4869. And what we saw is that in the control group, we had the same improvement in ejection fraction that we always see after a month because these cells were bioactive. But then when we destroyed the ability of the cell to make exosomes with the GW compound, we actually abrogated the effect. Okay, so now we know. Actually, exosomes drive the bioactivity of our cells. And so we now have sort of this explosion of understanding of the mechanism of action of our cells, how they work, why they work. And this led to the publication of many papers. And this is just a highlight showing the headlines of several of the papers that have been published over the past few years as we dig into the understanding of how the exosomes are made, what they do, what their contents are. And I can truly say that we are one of the very few cell therapies now that have a deep understanding of the mechanism of action of our cells. We've actually identified 3 microRNAs of interest that we're using as our potency assay. But we also learned a lot of other things about the exosome along the journey. So now we are moving forward to think of exosomes as an independent product. We think, okay, we're going to use the CDC exosomes. And we, like others, as I mentioned earlier, think the eye is probably the best place to start with a new biologic. And so we use an injury of ocular burn in the right eye. The left eye is left intact. we inject exosomes into the eye, and then we study the animals by looking for biodistribution over the course of 72 hours. And this large graph highlights the amount in the right eye, the injured eye, over 1 hour. Look how very little is in the left eye. Now we understand this so much better. Exosomes track to the site of injury. And I'll talk about that in a minute with IV delivery. And then you also see them in the places you'd expect, the liver and the spleen, but wait, you see it in the heart. You see quite a bit in the heart. And then if you look 72 hours later, you still see some expression in the right eye where we had put the exosomes, or not expression, but actually the persistence of the exosomes. But look, 72 hours later, we're seeing them in the heart. Now, I should have mentioned that our cells, the CDCs, the cardiosphere-derived cells, come from the heart. These exosomes then are coming from heart cells. They know to go home to the heart. This gave us the idea, okay, we can actually target these. If not nature's allowing targeting, we can augment that. And then we also went forward to see what happens with intravenous delivery, because as you remember, I mentioned earlier that sort of the gold standard where we really want to go with therapeutics is IV delivery or the ability to treat systemic diseases. And what we saw with the IV injection is that the cells are going, or the exosomes are going exactly where we'd expect them to go, again, the liver, the spleen. But this injury model, which was an injury model of skeletal muscle injury, is where we actually create a injury to the right leg and not the left leg. You're still seeing it go more to the right leg than to the left leg, but you're also seeing, again, expression in the heart. So these cells or exosomes definitely track to sites of injury. They track specifically to muscle and, of course, the normal filtration systems in the body. But what really then coalesced in our mind was the concept that now you could target the exosomes. And then finally, just finishing up the story of the CDC exosomes, what we found is that the contents of each exosome class are different depending upon the cell type we start with. So we did a direct comparison with the CDC exosomes, compared them to mesenchymal stem cell or MSC exosomes. And we found, and you can see it broken down in these pie charts here, that the CDC exosomes have a very different expression profile than the MSC exosomes. Now, what that means biologically, I could spend probably the next hour talking to you about what we've discovered and why we think CDC exosomes would be a better option than an MSC exosome. But what we really then came to realize in our sort of aha moment is that exosomes, as nature's delivery system, can be manipulated to be used specifically as such. So they can become an engineered opportunity to deliver, whether it be to treat an infectious disease using an mRNA or some other type of vaccine approach. We've actually seen the vaccines in the lipid nanoparticles. We think we're developing a Gen 2 vaccine housed in an exosome. In oncology, as I mentioned, siRNAs are being used currently. No reason why they wouldn't do better in an exosome. Cancer vaccines are one of the opportunities there as well. Monogenic diseases, this would be, I have sometimes joked that delivery of RNA using exosomes is poor man's gene therapy, because you don't have integration into the genome, so you don't have long-term expression. So people might have to come in once a month, once a quarter to get their mRNA exosome formulation in order to drive protein expression. But to save themselves from the downstream safety risk, I think most people would be willing to do that. So we are actively thinking about monogenic diseases using mRNA-loaded exosomes. And then, of course, inflammatory diseases, as I mentioned, exosomes naturally track to the site of injury, put something on the outside that tells it specifically where to go where the injury is occurring, load it with a protein or an mRNA, and you have a very powerful construct. Typically, exosomes have 3 extracellular markers or receptors: CD63, CD9, CD81. Different cell classes have different proportions of these markers, but what's most important is you can take advantage of any one of these, put a moiety on it, and use it for targeting or for sending the exosome exactly where you want it to go. Loading these are quite simple, actually, when you develop the methods as we have, and so you now have a great opportunity to use the exosome to treat a variety of diseases. So I alluded to this in the previous slide, but I just want to highlight that you can load the exosomes with RNAs. It allows for tunable regulation of gene expression. The three primary classes that we are working on right now are messenger RNAs, obviously for the uses that we've discussed, protein translation or transcription and translation of high fidelity proteins, siRNAs, these are small interfering RNAs. I've alluded to those in their use in oncology, and microRNAs, which are very small pieces of RNA that have a similar activity to the siRNAs. They tend to block transcription or translation, but they are modulators of bioactivity, and they can be very powerful in the use of the development of therapeutics. So any one of these classes can be safely and easily loaded into an exosome and then used to deliver to treat a variety of diseases. So how do you load an exosome? Well, there's two basic ways to do this, and then there's subcategories under each. The simplest way, but the way that sort of has the least control to outcome, is actually before you isolate out the exosome. So remember, these exosomes are naturally loaded. They take advantage of the materials that are in the environment. So you co-incubate a cell and whatever molecule of interest you have, and you look for them in your exosome. The transfection efficiency is usually very, very high, and so it's a relatively easy way of deriving an exosome. What's even easier, and the way that we actually are pursuing it most actively right now, is direct mixing. So you take the exosomes out of your cell. You can use a standard cell type. We're using a very simple, commercially available cell type for most of our studies. You take your RNA of interest, you co-incubate them or mix them together. Sort of, if you can sort of think about an oil and vinegar kind of response, shake it up a little bit very gently. And then you can get the contents to be taken up by the exosomes for use in a variety of clinical uses, or you can use sonication or other ways of permeating the exosomal membrane to allow the contents, the RNAs to slip inside. This is better, obviously, with a small RNA, like an siRNA or a microRNA. Again, this is very simple and time-saving. Electroporation is the way that we are pursuing it at this point as well. Again, it's a way of sort of just shaking open that membrane and allowing the contents to slip inside. So just like some of the other players in the space that are looking to show that you can drive mRNA expression, we decided to use as our seminal candidate for Exosome Therapeutics the development of a vaccine. Because of the temporal relevance of COVID-19, we decided to build an exosome-based vaccine for COVID-19. We decided to make ours a little bit different than that which is out there, not just using the spike protein, but using the N protein as well, which is one of the highly conserved proteins. There's obviously a lot of benefit potentially in terms of side effects, efficacy, dosing of using an exosome-based RNA vaccine that we are very excited about. So the first thing we had to do was show that the mRNA could be delivered in vitro or in vivo, not just in vitro. We started with a fluorescent protein called Antares. Antares we're using the mRNA version. We inject the mRNA into mice, which has been preloaded into the exosome. So it's Antares into the exosomes, and then that formulation intramuscular into the right flank of the mouse. You can then use a enzyme called DTX, which you inject into the mice. It cleaves off an end of the now expressed protein, which allows it to light up fluorescently, and we can image that using an IVUS system, which is shown here. And what you can see is that 24 hours, 48 hours, and 72 hours, even if you turn up the background as high as you can, you're still seeing far more protein expression, or actually real protein expression, not just noise, in the animals that were treated with the exosome Antares construct, as opposed to those that were just given the DTX injection, so no exosomes with nothing loaded inside of them. So this gave us very good indication that we were actually able to load an exosome with an mRNA. we were able to inject it inside of the mouse, and then we were able to see high fidelity protein translation, which was then bioactive because it was able to be cleaved off by the enzyme. So all of this said, okay, this process is actually working. Because we're developing a clinical product, we wanted to make sure there was also clearance. This is a study that was done and submitted with our package to the FDA, showing that we have complete clearance after just several days of the formulation, the vaccine-based exosome plus mRNA. This then shows that it's very safe as a delivery method. The exosomes go away. Now, we also wanted to compare them to lipid nanoparticles. If they weren't as good, or if lipid nanoparticles were better, we sort of had wasted our time running to this gate. So we did a study where we took simple lipid nanoparticles and loaded them with the mRNA that we were using. And then we also took exosomes loaded with the mRNA, same mRNA, experimental mRNA. And we looked at two basic parameters. We looked at body weight and we looked at histology, so inflammation inside. Body weight in mice, as in people, is sort of a reflection of general well-being. You can see in the control animals, we see the body weight is completely stable 3 days after injection with the vaccine construct. And so they just got the open exosome with nothing loaded inside, so just pure exosomes. Then we have the formulation where the animals were injected with the exosomes with the mRNA. And you can see that their body weight is very stable across 3 days. But in the animals that had just the lipid nanoparticle plus the mRNA, so analogous to the vaccines that are commercially available now, you can see the animals had a statistically significant drop in body weight over the course of 3 days, suggesting they weren't feeling well, which for any of us, including myself, that have had the vaccine, it can leave you feeling pretty crappy for a few days. So this is in fact a reflection that the exosomes seem to be less toxic, at least in terms of driving side effects. We then did a full study histologically. This is shown H&E, or hematoxylin and eosin staining of a variety of organs taken after injection with our vaccine construct. And we again did the same groups control, the empty exosome plus the formulation, lipid nanoparticle plus the formulation, which are the mRNAs. And what I'm highlighting here is that you see inflammation in the spleen of the animals when you compare them to the exosome group or the control group. So we now have two schools of evidence that we see potential side effects of the lipid nanoparticles that we don't see with the exosome. So this has obviously spurred us even faster forward in our development of an exosome-based therapeutic, and in this case, a vaccine. So I'd just like to summarize before I share with you some of our data, the use of exosomes for drug delivery. They can deliver a variety of therapeutic cargoes. We right now are focusing on mRNA, but we're also working to develop siRNAs and some of the other non-coding RNAs, lipids, proteins, and other compounds. Even small molecules can be used inside of an exosome. There's a variety of ways of getting the contents inside the exosome. A lot depends on the size of your construct, how you do it. mRNA is the biggest, and so that requires some different types of modifications than an siRNA or a microRNA. We talked a lot about the administration routes, and we talked about the opportunity for targeting strategies. I spent less time on this because this is a little bit farther behind in terms of its clinical development, but basically people, including us, are using the opportunities provided by the natural receptors on the exosomes, which I spoke of, to put targeting moieties on the outside. So the exosomes, as we see them, will be the future of delivery of nucleic acids, or pretty much anything you want to get past the cell membrane safely without integration into the host genome. So now let me share with you a little bit of the data that we have collected based on our development of a vaccine for COVID-19. This flow cytometry analysis shows that we have a 90% purity of our exosomes once we've done the isolation of them from our cells, and we're using HEK293 cells, which have high expression of CD81. So basically the takeaway message from this slide is we have a pure population of exosomes. We're developing a bivalent mRNA plus exosome vaccine. I spoke about that a moment ago. We're using the spike protein as well as the N or nucleic capsid. We believe it will help drive not only an antibody response, but a T cell response, and ultimately also provide for longer term activity or immune protection. The exosomes, as I mentioned earlier, are potentially superior to the liposomes or the lipid nanoparticles because they're not recognized as foreign by the immune system. You can get the payload directly to the cytoplasm. You don't have to worry that the lipid nanoparticle happens to bump into the cell and delivers its content. These are directly going to cells. And as I mentioned earlier, biology shows that exosomes target the site of injury, not important in a vaccine because there is no injury, obviously, but in therapeutic development. And then the platform is going to be developed to use exosomes as delivery vehicles for other targeting opportunities or disease processes, which is very exciting. And I'll share with you a little bit about that at the very end. This set of studies were very important because we were able to show that we could have high encapsulation efficiency, greater than 90% in some cases, but certainly between 70 and 90 of our S and N mRNAs into the exosomes. And what's very important and shown in this Western blot or protein expression profiling on the right here is that when you look at the intact exosome plus mRNA, so you have the S and the N inside the exosome based on this encapsulation efficiency I showed you over here, you see absolutely no expression of the S or the N protein. We know that because here are the controls for the S2P and the N protein that we're using here. These are blank and open, so we know what the proteins look like. We don't see it here. But if you lyse the construct, if you actually break open the exosomes, then you are seeing the S and the N protein. And again, this shows that we have very high encapsulation efficiency, and the contents, the mRNA, is highly protected by the exosome from degradation or breakdown, showing that this makes a very potentially useful biologic product. This is one of my favorite graphics because it's sort of the evidence that a picture paints 1,000 words. So we did electron microscopy looking at the formulation. And what this shows, and you can see here when we see the measurements, this hexagonal shape here is actually free lipids. So we use a little bit of lipid loading reagent to encourage the mRNA into the exosome. And then what you can see here, you can see very clearly the lipid bilayer membrane, which is really exciting. You can't see the contents inside, but the reason we show this highly magnified version is that it's swelled from the basic exosome shape that you've seen up here, for instance. And so we know that it is actually loaded with contents. We're assuming it's the mRNA since it's the only thing that we put in there. And then here again, we see the loaded mRNA with the lipid nanoparticle. You can kind of see that the lipid nanoparticle is sitting free here and about to help integrate into the exosome with potential mRNA inside. So this gives us very good indication that our strategy of loading the exosomes with mRNA is working. And then finally, we show that the vaccine is potent when we are able to look directly at the expression of the protein of the N and the S. We see a really good protein expression. So we treat, we look at the expression of the protein, run it out on a gel, and we're able to see that we have expression of the spike and the nucleocapsid, suggesting again that we have biologically active product. And then finally, the proof of the pudding is in the animal studies. This is some of the preclinical work that we have shown to FDA has been published both in bioRxiv as shown here and now recently in the Journal of Biological Chemistry, where we show long-lasting antibody development to anti-N or the nucleocapsid and the anti-S. This is going out to 84 days, and we see very statistically significant improvement, or not improvement, but expression of the proteins, suggesting a long-term activation and protection for the immune system. We also show a very robust T cell response, as you remember or know. The T cell response is the memory response. It's actually what drives us to be able to chase invaders out of our body very efficiently, and is therefore very important, and could also drive long-term efficacy of the vaccine. And I think, as people realize now, the efficacy of the vaccines that are currently available may be waning. We may be in a situation where we need to get boosted as frequently as every 6 months. We haven't tested our construct in people yet, but we're hoping that we'll be able to extend the time at which the vaccine is effective based on this very robust memory response to both the nucleocapsid and the spike protein. And please excuse the spelling error, nucleocapsid. So I think there has been a global concern over the fact that some proportion of people, primarily young men under the age of 30, develop a myocarditis post-vaccine. And in fact, some countries have actually suggested that those people do not get vaccinated. Some countries have even prevented that vaccine from moving forward in that patient or people because of the potential impact of the myocarditis. What we were able to see, and this was actually a serendipitous discovery, is as we were looking at the histological implications of our vaccine, we were able to note that the control group, the lipid nanoparticle that we use, lipofectamine and mRNA, led to actual injury on the heart. This is shown right here, and you can see some fibrosis or calcification shown on the surface of these hearts, which we saw in 6 of the 10 hearts treated with this construct. We saw it in 1 heart that was treated with the exosomes plus the S plus N, and 1 of the control hearts. So this suggests that perhaps something is going on here. We're deeply involved now looking at the histology of these hearts and other groups of these hearts to see if this manifestation occurs more frequently, but it certainly suggests that a lipid nanoparticle is more toxic than the exosome, and therefore we think the exosome can make a very, very interesting vaccine candidate loaded with mRNA moving forward, and also for therapeutic opportunities as well. So what problems can be addressed in addition to vaccinology using exosomes? Well, I've spoken of them all here today, but I'll conclude with a reminder that they can be used instead of viral vector delivery. An exosome can be loaded with mRNA. You have to deliver more frequently, obviously, because you don't have long-term integration into the genome, but the safety implications obviously are suggestive that an exosome-based system with mRNA could be a much better opportunity than traditional gene therapy offers, or at least until we get on top of some of the safety impacts. We can use RNAs to address whether it be monogenic diseases, infectious diseases, oncology. And what we know is that synthetic nanoparticles have not been very successful as delivery vehicles. It works fine for a vaccine, and maybe even the immune response that they generate independently could be a good thing, but you certainly wouldn't want somebody who's coming in every month for treatment of a monogenic disease for them to be laid low for several days because they don't feel well based on the toxicity of the lipid nanoparticle. So we are moving forward actively with the development of exosomes as nature's delivery vehicle with the platform Therapeutics, starting with our mRNA vaccine and other products following behind in the pipeline. I'd like to say thank you to TriLink. TriLink has made so much of our work and others' work possible by the modifications that they've made to the RNA, the extension of the stability of the RNA, and a variety of the work that's been done. So thank you, TriLink. Thank you for working directly with Capricor, and we look forward to long partnerships as we ultimately change the way medicine is practiced. Thank you.
Thank you so much, Linda. What a great way to kick off mRNA Day here. Linda, let's squeeze in a couple of questions. Had a lot of audience questions for you. So this will, let me just dive right in. What is the source of the microRNAs that you load into the exosomes? Are they synthetic? And how do you fully characterize the resulting exosomes?
So we started in the microRNA discovery pathway by looking at the ones that were in the CDC exosomes. And we have several that we've published on that seem to drive the biology of the cells. And again, we're using them as potency assays. In terms of the loading of the microRNAs and using them therapeutically, we haven't disclosed that yet. And as a representative of a public company, I can't really say exactly what we're doing specifically, but I will say that ultimately they will be synthetic and that they will drive some type of biologic response potentially in a oncology-based disease.
Can you say anything more about the loading efficiency compared to LNPs?
Yeah, so the LNP loading efficiency is obviously very high. It's the toxic impacts of LNPs that we're trying to get away from. So we're not trying to compete with loading efficiency. What we are delighted with is the loading efficiency into the exosome. So this was considered one of the great barriers to entry. I think a lot of groups believe that exosomes are better delivery systems than a lipid nanoparticle, but to be able to derive greater than 90% loading efficiency was really something that we were excited about. And these are with mRNAs, and they're not simple mRNAs, and they're not specifically small. So this gives us a great hope for moving forward with this loading strategy.
Great. A couple more. What is the potential of, how scalable is this technology compared to LNP production?
Right. So, it's not an apples to apples comparison. Obviously, you can make vats of LNPs probably easier than you can make exosomes, but that's not the issue. The issue is if you can make a better product, then you do it, and you have to, of course, do it in a way that's efficient. We believe we have scale-up technology in our hands that's going to drive that very efficiently, and we do not see scaling up the technology as a barrier to entry. So it's not going to be, a highly curated type of technology.
And our final audience question, what is the potential of microRNA, Linda, as a therapeutic for diabetic microvascular complications like diabetic nephropathy?
So sure. So diabetic nephropathy is one of the interesting issues that could be addressed with a microRNA. And obviously kidney disease is one that would be of great interest in targeting. It's an area that is of great interest to us and others. So we certainly are hopeful that we'll be able to attack diseases like diabetic nephropathy, diabetes in general, but stay tuned to the announcement of our pipeline as we move forward most expeditiously.
Okay, Linda, fantastic. Let's leave it there. Linda Marbán from Capricorn Therapeutics. Thank you so much.
Thank you.